All right, yeah, I gotta prove you guys are real to the teacher. So do something, react to me. She can barely see you.
I am a real human, not a bot.
All right, this is not a pre-recording. That looks.
Ask the question, Brian.
All right, so basically, I'm gonna talk about CRISPR and how it's clustered. It's basically just clustered, regularly interspaced, short palindromic repeats and its effects on genetic engineering. Okay. So first, genetic engineering, because you guys have never heard of that before. So genetic engineering is like it alters the DNA makeup of an organism, so you can alter or delete or add new segments of DNA. And it's used in a bunch of things, but it can primarily be used in like production of cancer therapies, brewing yeasts, or genetically modifying plants and animals. It's also pretty important to a bunch of things, but right now it's used for increased crop yield or more resilient food. I'm sure if you go to like Fry's or Bash's or Walmart, you can look at the bananas and tomatoes there. Odds are those are genetically modified to look bigger or to taste better and whatnot. But in other cases, it can restore biological diversity to environments or just manage invasive species. And CRISPR, it's an immune system. It's similar to ours. Microbes use CRISPR to identify and eliminate viruses. It was discovered in 1987 by Ishino in Osaka University, Japan, and its first major breakthrough occurred in 2005. And a basic summary of how CRISPR works is when a virus invader, enters the bacterial cell, the bacterium will absorb some of the invader's DNA into itself, and then when the exact same virus comes in later to invade it, it will instantly find it and eliminate it because it literally absorbed it into itself. But a more in-depth version of how CRISPR works is, like I said, it's similar to the human immune system. When a virus infects us, we create immune memories. They're in the form of antibodies, and when we're infected by the same virus later, they will quickly recognize and then destroy the virus. What CRISPR does is it creates a genetic memory, and when a cell is infected, CRISPR creates the genetic memory of the virus, so the bacterium can insert the virus's genome into itself, and then CRISPR will use that new DNA to form a guide RNA for the cell. The guide RNA is basically just when the same virus infects that cell later on, it will kind of guide the cell's quote-unquote immune system, and it will quickly recognize and neutralize the threat with the previous genome it had. But yeah, there's some advantages of CRISPR, like precision gene editing. It's stupidly precise. It's much more precise than any other tool we have access to, and by many times, so it makes it a lot safer and accurate. It's a lot faster. Previously, it took a complete sequence of human genome. It took 5 years to generate, but now it can take less than 24 hours with CRISPR, or at least like 3 days at most. But another main reason is it's RNA coded rather than protein coded. So basically, protein redesigning is incredibly tedious, it's annoying, it's time consuming, it puts a burden on bioengineers. But CRISPR using RNA to a bioengineer, it's incredibly simple to redesign, and it's just kind of like operating a GPS where you want to go to point A, you put in some coordinates. If you want to go to point B, you just change the coordinates, and CRISPR will guide you there. That's basically how it works to them, and that's why they love it. They love it so much. So yeah, there are some medical applications like the treatment of genetic diseases. So genetic disorders aren't really incurable anymore. Right now, diseases such as like anemia or cystic fibrosis and muscular dystrophy are either like mitigated or corrected. Genome studying is much faster. I already said this, but 5 years of studying to generate a human genome was completed in 24 hours. And also stem cell research. So CRISPR can help advance stem cell research, which can then be used to create regenerative medicine, so it can repair or replace damaged tissue. It has some agricultural applications like depolyamerase, which disassembles the cell wall of fruit when it softens. RNA can interfere with the production of depolyamerase, which can make a tomato have a longer shelf life and become firmer, but it won't make it taste bad or influence its nutrition. It can also make fruits that have abundant nutrients like carotenoid, GABA, iron, and zinc contents. Those can be increased in their respective fruits, and it can improve flavor. So when they learned that BADH can convert gabald into 2AP, they would just mess around with the mutated strands of BADH2, leaving only good BADH2 to convert that stuff into 2AP, and it would just make food taste better. But, sadly, there are ethical considerations. So before a baby's completely born, you can just alter how it comes out. So maybe you want your child to not be a dwarf, so you can make them come out tall, or you can make them blonde over black hair, or maybe you hate brown-eyed people, so you can make them have blue eyes. So that can be a bit odd. You can also just choose to make them hideous, so there's that. There are ecological changes, like certain pests that we really don't like mosquitoes, could just be erased off the planet, which could be good. But if we could erase a pest such as insane as mosquitoes by making our blood lethal to it or something, or livestock lethal to it or whatever, we can probably do that with literally any other species on this planet. So wouldn't it be great? And we could also just create better humans. So like everything, it would cost money. It would probably be expensive. There are a lot of poor people on this planet. If you want to create a human that's faster and stronger, only a select few would be able to afford it, and it would probably amplify the difference in socioeconomic status even further, because not only are they rich, they're probably fit and buff. And that's not very cool sometimes. So yeah, in conclusion, CRISPR is a pretty incredible tool if utilized correctly. And like everything, we can also weaponize it and create horrible horrors beyond our comprehension. But as long as we develop it responsibly and, safely over the next few years, it can be a tool to make humanity prosper. And we're excited.